Okay, thank you. Thank you. Can we have the speakers come up? We went a little bit long, and we only have about 15 minutes left for questions and discussion. But while the speakers are coming up, I note that we have all academics here, so I look forward to participation from non-academics, especially pharma industry in the audience. And then I'd also like to try to focus us on what we saw was a lot of sort of basic science data or a lot of data that was not yet practically useful in the clinic, with the exception of TMB, which has already been to some extent adopted. So I'd like to also address the topic of how we take these things from as far as the microbiome or some of the things that Dr. Azar talked about into actual companion diagnostic tests. Do you want to start? I think we have a question to start us off, or should we address those issues? No?
Whatever you'd like to do.
What? Go ahead. You can start off.
Okay. Good afternoon or good morning. Good afternoon. My name is Ron Klein. I'm from CMS, and I appreciate the lovely science that was presented. I'm going to ask a health policy question that has no answer, which is like most health policy questions, probably best directed to Dr. Rim and to Dr. Shelsky. So all the data that was presented show a continuum of responses in biomarkers. There's very little 0 or 100. And so obviously how you define a positive response or a negative response has not only tremendous clinical implications, but probably tens of millions of dollars and hundreds of millions of dollars are riding on that, as we sort of noted from the ASCO session this year. And as you pointed out, I think both Dr. Shilsky and Dr. Rim, there are a multitude of different tests that have various different sensitivities and specificities. So the question from a health policymaker standpoint would be, how do you define as scientists what's positive, what's negative, and how do you create a framework that allows us to rationally look at different test results?
Well, I'll take a shot at it first, and then Dr. Shelsky can. So it's a great question, because at what point do you introduce a test into the clinic and kind of hope that it's good versus require a lot of evidence, and then a test may never make it to the clinic? And how do you decide at where you're going to place that cut point? And so what we've been doing, and I think as a good strategy to do, is to sort of look at the predictive value of the test. That's hard to do because it requires a no treatment arm. But when you have predictive value and you show an interaction, that can be a first, sort of proof of concept that this test has legs. And then the next thing we do is look at the area under the curve. And unfortunately, we didn't do that so much in the past, but it's catching on now. And I think that's a good way to evaluate the effectiveness of the tests and compare the tests between each other. Ultimately, the area under the curve can also define a cut point, because as you know, sometimes it's strategic to have high sensitivity and low specificity in order to include as many patients as possible, but you don't want to include too many or the test will be meaningless. In fact, we saw a bit of an illustration of that in the history of the PD-L1 testing. So I would say that the two critical points for us are predictive value, using a no treatment arm, or what we're trying to induce is, the problem with predictive value is you need to have tissue from the patients who are on a control arm. And now there will never be another control arm in melanoma that for patients who don't get a PD-1 axis drug. So does that mean we can never have a future predictive value? No, I think we need to meet with our biostatisticians, and we actually have some ideas of ways to address that issue. But predictive value when you can, and then a large area under the curve.
I don't know that I have a lot to add, certainly not on the analytical validation front. But I do think, Ron, that the broader policy issue again comes down to life cycle learning. When do you have sufficient evidence about the predictive value of an assay, or even the utility of a drug, to allow it to be introduced into the marketplace, where you have a certain level of confidence that, say, in the test of the biomarker test, that it actually can identify a population of patients likely to benefit, recognizing that every test is always going to be imperfect. And once you're in the market, you still have a lot to learn, because typically you have much broader use, much more heterogeneous populations, knockoffs on the test and so on. So I think it sort of comes down to a question of for each decision maker, whether you're a regulator, a payer, a clinician who's determining whether to use a test or a drug, we have to have a certain threshold to say, this is enough evidence to allow me to employ this test or this drug or to approve it or to pay for it, recognizing that the evidence is still incomplete. And then we have to continue to gather the evidence and be prepared to make modifications to our initial decisions based upon the accruing evidence.
Thank you.
Dan
Chen, Genentech. Two quick comments. So the first one just relates to I'm interested in the fact that when we look at these presentations, a lot of the assessment and even cut points for the biomarkers that were discussed were made on things like IHC intensity or response rate. And today in 2018, we have a lot of survival data for these biomarkers. And so shouldn't we really be focusing on clinical utility on endpoints that are important to cancer immunotherapy, like survival, where maybe that's really where we should be focusing. And then the second was just around the topic of this entire meeting being combination therapy. So we heard a lot of interesting biomarker data as it relates to PD-L1 and PD-1 inhibitors, and that of course has been important for the development of PD-L1 and PD-1 inhibitors. But it's less clear how many of these biomarkers are actually going to have clinical utility for combination therapy, and that many of these biomarkers, whether it's microbiota, whether it's PD-L1 staining, whether it's tumor mutation burden, may be good biomarkers for benefit of cancer immunotherapy, but may not help us separate out which combination immunotherapy is best for a given patient.
And that's a great point, because I think, in fact, all the biomarker work sort of follows the clinical trials. And in fact, it's hard to make a biomarker for the combinations when the combinations are in phase 1. In fact, it's always hard to have any kind of biomarker in phase 1 because no one wants to eliminate patients who might respond when you've spent all this time with the new drug. So unfortunately, biomarkers tend to follow that. And in my introduction, as I said a little bit, I don't believe that we can really talk about biomarkers for combinations yet, even though arguably that's where we need them the most. We need the biomarkers to separate the patients. So we need to come up with new strategies to approach biomarkers when we're getting into the phase 2 and phase 3 so we can address that combination problem. Question?
Thank you. I just want to make one more comment related to that. So I think that the other thing that is helpful in that regard is if you've established a setting where there's clear single-agent activity of PD-1 blockade, then taking a step back and saying, what is the nature of the tumor microenvironment and what is the nature of the response to PD-1 blockade gives you, I think, important clues about possible rational combinations of therapies in that setting. Now, it doesn't guarantee they'll work, but it does give you real clues about the nature of the response, the expression of candidate targets, and some insights in terms of thinking about how to develop next step combination strategies. So it may not be that it gives you immediately biomarkers, but it definitely gives you insight into possible next step strategies.
Yeah, along those lines, I think that as far as TMB, there seems to be value with combination with PD-1 with CTLA-4, potentially with the chemotherapy combination. So I think that when we look back at all of our phase 1 studies where there's a lot of patients going on with immunologically less responsive tumors, and we're making assessments on whether these new drugs work or not. If we had a TMB threshold or some sort of threshold for patient selection, we actually could make some interpretation of the new immunotherapy agents better than having 2/3 of them being pancreas cancer, where you wouldn't necessarily expect LAG-3 or some other checkpoint therapy to be effective.
Thank you. I'm Türkan Gardiner, applied statistician. I really congratulate all of you for attempting a topic which is so relevant to precision medicine and personalized medicine. And I remembered a topic of a lecture given on personalized medicine, which said, getting the dose right. So aside from being a responder or non-responder, a certain dose might be too high for some patients or too low. And in dose titration, sometimes we start with the highest tolerable dose and go lower on successive trials. So as a statistician, I thought of a matrix. For a given individual, he or she may be responding positively to some medicine, but then another medicine might be antagonistic to that individual. So, or that medicine might not be at all effective for that individual. So I don't know how to answer the query. However, the personalized or precision medicine aspects should also be considered in the clinical trials for phase 2 or phase 3. Thank you.
I mean, I'm sure we would all agree with you, but I'm sure we would all agree with you. But I think in a sense you're articulating the fundamental conundrum that we all face, which is that we derive evidence from studies of populations. We have to make treatment decisions for individuals, and we're never sure exactly where that individual patient is in the population who was studied. That's used to derive the evidence. So that's I'm not sure that there is a simple solution to that, because each patient is unique, and the data that we derive from clinical trials is for a representative group of patients in a population. And the best we can do is to try to intuit how closely our particular patient fits with the characteristics of the population that was studied.
Last question. Last question, and then we will, I guess, move on to the next session, which is lunch, so it's always hard to be before that.
Then I'll try to be brief. Stan Gerson from the Case Comprehensive Cancer Center in Cleveland. This is for Richard. You sort of suggested a little bit of put on the brakes around the disease agnostic approval, which we now have, which is actually a provocative event in my mind for the FDA to do. And yet in each of the areas where we're looking at biomarkers as predictors, a portion, if not the majority of patients are non-responders. Are we going to get to the point where we'll need to retract away from disease agnostic approval because we can clearly define non-responders in a way that says those people should not be exposed?
I think that's a really key point. That is that how do we define the non-responders? Because in some way, it's a lot more important to define the non-responders than it is to define the responders. But the problem or the conundrum is that the responders are who needs to be defined to get the drug adopted in the first place. So there's a tension between the need to define the responders, which is the pharma companies, and the need to define the non-responders, which is the payers and the patients, arguably, who aren't going to benefit and shouldn't be subjected to the toxicities. And I think that now with the movement of immune therapies into the adjuvant setting, that's especially poignant or especially important, because there's real toxicity with immune therapy. Yes, it's better tolerated than some therapies, but there's real toxicities. And so now we have to figure out if we're going to have companion diagnostic tests that take us into figuring out who the non-responders are, whether they be MMR or TMB or whatever. I think we need to we need to really focus on that area. I'm interested in the other panelists.
Stan, I think you've you've hit on a key point, of course, and I think we have to be open to the notion that as we gain experience with using drugs in these histology agnostic settings, and hopefully we have a mechanism to gain that experience. I mean, one could argue, for example, that we should have a national registry of pembrolizumab use in all MSI high tumors, right? So that we actually know over time exactly how the drug is being used in practice, in which tumor types, with MSI status defined by which test, and we record those outcomes. And one, should be, potentially prepared to actually limit the label over time if it becomes clear that there are certain histologies, that actually don't benefit from that drug, even though they have MSI high tumors.
Okay, I should probably, one more question? Okay.
Sorry.
We have
I'm
to
sorry.
let,
I just want to bring in
Dr.
a
Nansen.
concept for you guys to think about. The major clinical trials are running their course. And if you buy into the concept that plateau is being reached, it seems pretty obvious that you could look at those patients who are plateauing to decide whether have they had enough therapy. In other words, define not just by responders, but by survival. Just a bit to Dan's point, how much could we learn about the existing clinical trial data to point out that, that patient group have gotten enough therapy, whether it's monotherapy, for melanoma, or combinations, or monotherapy for lung cancer. And that would at least then define the most successful population for which probably no more therapy is needed, and then segment the population going forward into let those folks get on, because they've got successful treatment available. Let's focus on those that don't actually get to that point, just for your consideration and comment.
Yeah, I certainly agree. I think that one of the tensions in doing that is that most of the tissue is gone then. That is not either gone or not available for use. That is, once we talk about 3 and 4 years out, and we really know who's succeeded in these trials, it's hard to then retrospectively obtain that tissue. And maybe that's a challenge to the people that do the trials, is to make sure they maintain that material so biomarkers can be discovered that really are entirely outcome-based as opposed to based on other correlative findings. I think I'm supposed to tell you where to go to lunch, but I don't know the answer to that question. So it's right behind. Apparently, it's right behind us. So thank you very much. Be back at 1:30.